Hello, everyone, and welcome to this Inside Scientific webinar titled Innovative Approaches to Tracking and Quantifying Behavior in Rodents. This webinar is sponsored by Noldis Information Technology, so a huge thank you to them for helping to make this event possible. Joining us today, we're very fortunate to have Dr. Jason Rogers, a senior research specialist at Noldis, and Devet Wolmarans, an associate professor at Northwest University in South Africa. They'll be providing an in-depth overview of two research protocols for tracking, quantifying, and analyzing behaviors in rodent models. And so with that, I'm very pleased to welcome our first speaker, Dr. Jason Rogers. Jason, take it away whenever you're ready.
All right, I appreciate that, Sarah. And everyone, good morning, good afternoon. Thank you for joining us. I'm very excited to present this webinar and talk a little bit about stress behavior on a novel elevated open field in long Evan rats. Before we get started, just a brief introduction of myself. I am a behavioral neuroscientist by training. I did a postdoc in neuropsychopharmacology, animal models of drug relapse, and that's going to be what I'm going to be talking a little bit about today. I've been with Noldis for a long time. I've done a little bit sales, services, consulting, marketing, etc. And I currently live in Ohio, in the Queen City of Cincinnati. So this morning, I guess today, I'm going to tell you a little bit about what we did, why we did it, talk about this novel elevated open field, talk a little bit about the tracking, and then some updated tracking with deep learning. And then, of course, show you what we found, and then we can get to some questions. So this project was part of a larger master's project that we did at MUSC. So what I'm presenting today is just the behavioral component, but all of this came from the research question of does stressing an animal, or do stressed animals self-administer more cocaine? And spoiler alert, because I'm not presenting these data, yes, they in fact do. So what we did is we had 16 long Evans rats. We implanted a jugular catheter. You'll see a picture of what that looks like in a second, but basically it allows them to self-administer drugs directly into the IV. We then split them up. So half of them received chronic unpredictable stress, which you can see an example in the right-hand side of STAR protocols, some restraint, some cold, not allowing them to sleep, cold swim, cage tilt, wet bedding, etc. These are all pretty standard protocols for chronic unpredictable stress. After that, we allowed them to self-administer, all animals allowed to self-administer cocaine for 14 days in a sound attenuation chamber. After that, they were taken off of the drug, put back into the chamber for withdrawal and extinction over the course of 10 days. Then we did some reinstatement testing, some various things to see how quickly or how much they would want to return to drug seeking. We then did some blood draws for corticosterone levels to verify that they were in fact stressed. And then we did the open field testing. So I'm not going to talk about any of that other than the behavioral component. So just take my word for it and believe me when I say that the stressed animals were in fact stressed. So the test that we ran them on, you can see in the bottom right-hand corner, is a elevated open field. This was a 122 centimeter round open field, a 4-foot board that was painted gray. It was elevated 1.5 meters above the ground. And as with most behavioral testing, space is tight. And so we actually set this on top of a trash can inside of a 8-arm water maze, and that's what you can see on the left-hand image of those things popping out. That's a water maze, or an 8-arm water maze. So we set it inside of that on top of a trash can. And what this is it's an anxiogenic, stressful environment where the animals aren't allowed to hide in the corner. There's no thymotaxis available. It's a round area. There's no walls. And so it promotes exploration while simultaneously giving them an area that they want to avoid. And you can see in the upper right-hand corner, there's an image. This is from Conduct Science, one of our maze partners. They have a new, what they call a 3D open field, which is very similar. Theirs, of course, has a netting allowing the animals to climb down, but it's all based on the idea of taking the traditional open field, removing walls, elevating it, so you're creating this hybrid open field elevated space. One of the reasons why we did this was because when you do drug addiction, drug relapse research, timing, of course, is very important. We want to test the animals in very specific times. And so 24 hours of withdrawal, 48 hours of withdrawal, 72 hours of withdrawal, all very different. And so when you do your behavioral battery, whether you put them in the open field or then the elevated plus, all of that can really affect the timing. So we wanted to create a hybrid to see, well, what if we just tested them kind of all at once to see what we were looking at? And in that regard, we were looking at the time spent in the center. So the animals were placed in the center. So we looked at the time they would spend in the center overall, how much time they spent along the edge, their latency to explore the edge, and then, of course, the overall distance travel and speed. Now, all of this was done automatically and tracked via EtherVision XT. So I want to show you a little bit about the tracking parameters. So first and foremost is, as I said, these data were collected a long time ago. So they were first created and analyzed in EtherVision 5, for those of you old enough to remember what that looked like. I went back and retracked them in the most current version, well, at the most current version as of last week, which was EtherVision 16. And I used the dynamic subtraction. And what it does is it takes a reference image and then it subtracts out the current frame of the video from that reference image. And you can see a picture of that in the left-hand corner of what that looks like. It is the animal. And then you have a yellow blob. That's the tracking of the animal. But the thing about that particular contour-based tracking, you can see in the right-hand side, that when the animal looks over the edge and has a head dip over the edge, the track loses the contrast because the program can no longer differentiate the animal from the background. And you can see the way that it cuts off along that edge, the way that it cuts off that nose point. So I went back and retracked it with the new deep learning algorithm that's in EtherVision 16. And what this does is it has an artificial neural network based, something that's way beyond even my understanding. But what it does is it's able to more accurately track the animals even when their nose goes over. So if you look at the left-hand side, that's that contour base that I just showed you. And on the right-hand side is using that deep learning. And even though the blob, you can see the yellow blob is cut off as the animal looks over the side, the deep learning is still able to accurately detect where that nose is, even when the animal is looking over the edge and the program can no longer differentiate animal from background. The advantage, of course, is that's going to give you more accurate data, going to be better able to accurately track where the nose point is to be able to look, and I'll show you a little bit more of that at the end. Okay. So we have stressed and non-stressed animals. We put them on an elevated open field, put them in the center, let them run around, tracked where they went, and then we tracked them with this deep learning. So what did we find? Well, as you can imagine, stressed animals don't go to the edge as much, which you can see on the left-hand side. They spent less time around the edge, excuse me, and they had a higher latency to first contact the edge. So they spent less time around the edge and they took them a lot longer, a little bit longer to go to the edge to explore. Likewise, they stay in the center longer and they maintain a higher distance from the edge, their average distance from the edge. So they're not going to the edge as long. There's a higher latency to get to the edge. They maintain a higher distance from the edge, and they're spending more time in the center. Now, finally, they show the same distance moved, so they show the same level of distance traveled, so it's not a motoric deficit or anything like that. They show the same velocity, so they're showing the same level of speed. So they're moving the same speed, they're moving the same distance. They're just not showing that exploration pattern. And here's where I'm going to make a plug for my next webinar, which will be next month in June, where we're going to be talking about data storytelling, using these principles of saying, instead of just showing one graph, describing the data in multiple procedures and telling a story with that. So that's my plug for my next webinar. Okay. Now, last thing I want to show you is, as I said, the deep learning is better accurate, or more accurate, better able to detect the nose as the animal looks over the edge. And you see that when you look at the frequency and the duration of the head dips. Head dip is when the animal looks over the edge and literally looks down over the edge of the maze. And it's a little bit less necessary to look at the comparison between the control and the stress, because I already told you that stressed animals spend less time at the edge. So it's not surprising that they have fewer head dips. Okay, what we're really looking at is the deep learning versus the dynamic subtraction. And what you can see is that the head dips have a fewer number from the deep learning, but a higher duration. What that means is, because it's able to keep that nose point as the animals look over the edge, you don't have that back and forth as it's trying to model the nose point as the animal moves back and forth. And you're able to accurately detect where the head is and keep it over the edge, looking at that head dip for longer. So even though the data don't change that much, they are more accurate, and you're better able to detect where the animal is at all times. So in conclusion, chronically stressed animals have a higher latency to explore the edge. They spend less time at the edge, maintain a higher distance from the edge, and they spend more time in the center. Of course, they have fewer head dips and spend less time looking over the edge as well. And then finally, as I mentioned, that either vision with the deep learning is more robust, more accurate for nose detection. And you can see comparison. The top is with the contour-based, the bottom is with that nose, with that deep learning. And you don't see a big difference, right? We're not talking about a significant difference. Either vision is still the best tracking software there is, even if you're using contour-based. But with deep learning, you're just getting that extra, more accurate detection, particularly with long Evans rats, particularly on environments where they're going to lose that contour base. And with that, if you have any questions, I'm happy to share these data with you. I'm happy to talk more about this. Please contact me, jason@noldis.com. And again, I'd like to thank you for coming. Thank you, Sarah, for having us. And I'm really, really excited for everyone to talk more and to hear from DeWitt. So thank you.
Great. All right. And so with that, I'm very pleased to welcome you, DeWitt. Take it away whenever you're ready.
Thank you, Sarah. Thank you to scientist.com, Insight Scientific, Jason, to you and Connie and the team at Noldis. I've been working with your software for a while now, and I promise you that this will not be a subjective talk, although we will discuss the subject of subjectivity quite a lot today. So instead of focusing on a single behavioral paradigm, I thought I would elucidate a bit more to those of you that are interested in animal behavior and that works in the field of especially preclinical psychiatric studies, neuroscience, etc., of the importance of really looking carefully at what your animals do in the laboratory and the importance of minorities. My name is Wolmarans. I'm from South Africa, at the southernmost tip of this dark continent. It's a good place to work in, and I hope that today I can really bring some food for thought to your tables. Just a brief bit of background. So the focus of my laboratory is on the naturalistic expression of abnormal behaviors in animals, and with the focus specifically on naturalistic. So most of us that are informed in preclinical research know that most of the models that we have to our disposal today are actually induced models of some sort of abnormal behavioral phenotype, whether it be a neurodegenerative type of disorder like Parkinson's, or whether it's a cognitively founded disorder like OCD. Much of those models are founded either in genetics or in some sort of behavioral training or pharmacological induction, which obviously makes it difficult, because if you want to ask etiological questions of how certain animals, and then if you translate that to clinical samples, humans, develop to present with certain abnormal behavioral phenotypes, it's really difficult to do in an induced model. And so my colleague at the University of Cape Town, Dan Stein, came across an animal species, quite ironically, in the United States about 17 years ago, which is the deer mice. And I think many of you would be familiar with the species. So the behavior that he saw in those animals at the lab visit that he conducted at the time reminded him quite of compulsivity, in the sense that it was repetitive and it seemed just very purposeless for an animal to express such high intensity stereotypes at the time. So we found back to Potchefstroom here in South Africa, and he asked us whether we would be interested in looking in this behavior, and the rest is history. So that was about 15 years ago. And as I become older day by day, it becomes a hard fact of life that you actually start to realize that the older you get, you can actually spend a whole career just asking whether something is actually abnormal or whether you are just being subjective. And that's basically what we are aiming to do. So at the time when we started with this work, we wanted to develop this model as a framework for obsessive compulsive disorder. That was 15 years ago. It was high fashion to model diseases in animals. The whole notion changed now. So we came to realize that we will never actually do that in animals, and we must rather look for processes that are akin to some sort of dysfunction that might be involved in human disorders. So at the time when we started to develop this model as a model of compulsivity, we really did a lot of work, and the model went through a whole lot of rethinking and reconceptualization against the background of compulsivity. And what we came to realize is that there are animals within that population. So when you house them, a subpopulation of these deer mice, irrespective of sex, begin to present with abnormally persistent and repetitive and needless phenotypes. And the value of highlighting what really is abnormal against what is quite subjectively, I must add, normal, became really important when you wanted to ask questions about compulsivity. So I'm today going to focus a little bit on how we came to the realization that actually looking at your minorities is actually very important, and that in animal research, especially looking at naturalistic behaviors that's non-induced, that is very, very important to subjectively look at your data. So in order to be quite objective, one needs to realize what really is abnormal in animal behavior. Because once you did that, you can actually start now to investigate, but how did certain animals get to a point that they express such behaviors compared to other animals that remain quite neurodevelopmentally resilient and just become normal geriatric adults, if I can say it like that. So at the time, what we did is we looked at stereotyping, and I'm quickly going to show you 2 videos of what this behavior looks like. And we started to develop that against the background of compulsivity. Now, on your next screen, you will see 2 videos playing. On the left, you will have a repetitive jumper. So that animal will go the whole night like that. And I will show you just in the following slide what the data actually looks like. So what you see on your screen is 2 animals that I, at the time when I did my MSc degree here at pharmacology in Potchefstroom, actually described and told my supervisor this is abnormal. So on the left, you've got a repetitive jumper, and on the right, you've got a pattern runner. So these animals go in circles the whole night. The one in the left chooses a corner or 2, and then he starts to jump. So this apparatus that you see here was actually fabricated in Ohio way back in the 80s. It was one of the first infrared beam behavioral analyzers. But the difficulty with this was that we couldn't classify the behavior across a whole dark cycle. So what my predecessor did, the postdoc that did his work here before me, is we placed these animals in this apparatus for an hour. So when I did my master's degree, we quickly came to realize that when we handle deer mice in general for an hour, they become quite active, and that these stereotypes might be a false representation of what we actually see. So subsequently, I started to look at whole night behavior in these animals. Now, Sarah reminded me quite often that I shouldn't point on my screen because you can't see my pointer. So I'm quickly going to just explain to you what you are looking at. So in order to classify stereotypy as a compulsive-like behavior way back when, we looked at the DSM criteria of compulsivity quite obviously and came to realize that it's intrusive, it's repetitive, it's time-consuming, and it really interferes with the normal behavior of human beings on a daily basis. So I simply asked the question, will this be the case in deer mice as well? So what we did is we placed them for repeated dark cycles in a novel infrared behavioral tracker that was actually able to dispense food and water throughout the whole dark cycle. And what you see on the left axis of these two figures, on the left-hand side, again, you've got data for the jumpers, and on the right-hand side, you've got data for the pattern runners, okay? So it's the same animals. It's not necessarily the same animals of which the sex symbols are indicated, but it's the same study population. So we took 50 or 60 mice, I can't remember, and I'm not going to count now on the screen. But basically, we exposed them to this apparatus for 3 consecutive dark cycles, because that was quite important for me. A compulsivity is actually not a single day disorder. It's repetitive over different days. On the y-axis, you've got the number of beam breaks on the vertical axis. So that would be your jumpers. And that's the number of beam breaks recorded within a half an hour. So the values on the y-axis represent the average of the highest intervals that these animals actually generated beam breaks. So if an animal averages at 6,000, he had at least 3 30-minute intervals in a single dark cycle in which he expressed about 6,000 jumps. On the right-hand side, you've got a lower value because it takes a longer time for animals to go around in circles, and hence the y-axis scale is a bit smaller. On the x-axis, and this is a principle we will discuss throughout the next few minutes, so I think if we are all on the same page from here, the rest of the talk would make much more sense. So on the x-axis, you've got the time that they engaged in high stereotypical behavior. So we set a certain cutoff. I think it's 2,000 beam breaks for vertical activity and 200 revolutions within a half an hour for pattern running. And what you see on this figure, in the top right-hand quarter of each figure, is there's a number of animals that actually persistently engages in high stereotypical activity. So it's not a thing that it's only 3 or 4 30-minute bouts of the dark cycle. It can actually become as high as 30 to 80% of the dark cycle. Now, bear in mind that these are normal mice. We don't do anything to them. They get the normal rodent chow, the normal water, so they have probably the normal abundance of energy compared to the rest of the population. And that's basically how we discern between an animal that presents with now, what I like to call it, persistent behavioral phenotypes, and those that don't. The dark dots on the left-hand side of the figures are animals that never showed any bout of high stereotypical activity at all. Then part of my PhD was to ascertain whether deer mice, as with humans with compulsivity, would present with other forms of compulsive-like behavior as well, or and please, it's in other forms between parentheses, because I don't know whether stereotypy is actually a form of compulsivity. I don't know it yet. We are still busy with it 15 years later. But there were two tests at the time that were quite widely advocated, and which we tried to replicate in these animals, and that was the marble burying test and the nest building test. So I asked my supervisor at the time, Prof. Brian Harvey, whether he would be interested for me to look into different compulsive-like phenotypes, and bless him, he was amenable. Now, those of you that worked with the marble burying test before might know how subjective of a test that might be. When we tried to replicate results from many other laboratories, we failed simply for the reason that the second picture from the left, I know it's small, but that's your normal coarse sawdust. And these animals were so active that by merely moving in this arena, the marble settled to the floor of the testing cage. So it became very difficult to actually assess burying behavior. So we reinvented the wheel and we started over, and I tested different nesting and burying substrates. So on the right-hand side, there's fine river sand, and that was actually the substrate that we came to use because it's dense, it's heavy. So when animals want to bury the marbles, they really need to go for it. It won't simply settle to the bottom of the floor. Remarkably, what we saw was when you did the marble burying test in that way, then really all animals seem to bury marbles. And that it's not a question really of seeing group differences. Now, again, remember at this point, I wanted to see whether the same animals that also presented with jumping and running would likely also present with other phenotypes. So the black dots was the high stereotypical jumpers and runners, and the white dots was the normal animals. And then I want your eye to follow the far right-hand side of this figure, the far lower quarter of the y-axis, and from the combined average of marbles buried on the right hand. And you will see 4 animals indicated with triangles there. From the 30 or 40 animals in this figure, ladies and gents, it was only those 4 animals that we could classify as high marble burying animals based on the data. So I never looked at the marble burying test results in literature quite the same way again, because in our population, only about 11% of these animals, some were high stereotypical and some were low stereotypical, came to express high burying activity. So how we did this was over 3 nights. So we took a combined average of the number of marbles buried, and we gave them 15 marbles, which we spaced 3 by 5 in the normal home cage setup. We habituated the animals beforehand on the river sand so that the burying substrate will not be novel. So these 4 animals actually persistently engaged with marbles, and they continuously buried these marbles until there was nothing left of them. The other animals on the figure showed a moderate interest in the marbles, scratched a little bit and walked away. And I'll get to the importance of these 4 animals, because we had quite a come and go with the reviewer about, but so what? Why would you look at these 4 animals only? What does it say? You've got outliers in any investigation. That may be the case. But the question begs as to why certain animals present with such behavior. It might be outliers, but the question remains as to why. The second test that we looked at was nest building, and we assessed all these animals with cotton wool over 7 days. The 7 days in this case simply came from literature from a lab in Alaska of Abel Bultito, and we replicated their behavior because it was described in literature in a quite robust way, and we found the paper quite informative. So I asked my supervisor at the time, let's replicate a method in another species and see if we can build on literature. And what we came to find again was, irrespective of stereotypical cohort, that some animals on the left-hand side will build what we now today, I think it's about 11 years later nearly, we today say this is quite normal. The majority of the population would express behavior like this, while about 30% of DMIs would express behavior on the right. And that you can see in this figure. So once again, the total nesting score after 1 week, and then on the y-axis and then on the x-axis, you've got the variance in the behavior. So we are not looking for animals that build a big nest in one night only. You look for animals that build big nests in all of the nights, because that speaks more conceptually to what compulsivity in essence would be. So again, about 7 or 8 animals from the 30 animals, or whatever the count was we used at the time, came to express large nest building behavior. So that brings me by my next slide. And I really want to ask the question, so what does it all mean? Why do we look at minorities in naturalistic behavior? And why is it important to ask yourself subjectively what you feel is abnormal or not? And the reason simply is that the minority of animals actually reveal the majority of the findings. If you look at it from a perspective of psychiatry, what we normally do is we pull animals in a big group and we give an animal a specific intervention, and we compare that with a control intervention, and later you end up with large standard deviations and say, no, there's a big difference between these groups, or no, there's not. And what we came to realize over time, like many other scientists, I'm sure, is that behavioral data actually varies quite a lot. So if you don't separate objectively by looking with subject at your data, then you won't necessarily answer the questions that you feel you wanted to answer. And I'm going to show you a few examples. So what we did now was show you how we actually select animals that we feel is different. So in the next few slides, I'm going to show you what happens when you select those animals and you apply them in behavioral tests with novel questions about the cognition and the biology. The first major breakthrough that we had with this behavior was that I asked at the time whether, like in humans with psychiatric disorders that become marginalized and they kind of are perceived as different by their peers, just for the fun of it, let's see whether normal animals would actually perceive high stereotypical animals that we now selected based on our own criteria and saying that's different. So now we put normal animals in the same cage as a high stereotypical animal, and this social paper only focused on stereotypy, and see what happens. Remarkably, we found interesting data, and this was the first time that we had to acquire a program like EthoVision, since it was 3 animals per arena. We had different phenotypes in each arena, and we had to mark them with color markers and tell the software which animal is actually which animal. Now, ladies and gents, bear in mind that these animals are so hyperactive that you cannot place them in a normal social interaction paradigm and think you will get realistic data by sitting with a stopwatch and counting. It's not happening. So I'm going to explain this figure quickly, because in my view, I don't think my psychiatry friends feel the same. But in my view, this was the first findings that pointed to a cognitive involvement in these animals, and that really founded the whole foundation of the work that we are doing at the moment. So the 2 white bars on your left that says normal, 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 or high stereotypical, high stereotypical, high stereotypical, was 2 setups in which we had animals of the same phenotype. And we saw a remarkable difference in the duration that these guys wanted to spend together. So much so that the high stereotypical animals was much more sociable than the normal oaks, which wants to tell me something of society. The two black bars in the middle is actually two setups. The left one, the smaller one, was two normal stereotypical animals that we placed into a cage with a high stereotypical animal. And in that sense, the behavior indicated on that bar is how the normal animals actually approached one another, the time they spent with one another in the presence of a high stereotypical animal. We didn't see much difference compared to the first control. In contrast with this group, the high stereotypical animals actually again spent more time with one another in the presence of an hen animal. The most striking finding of the study is indicated in the two bars with the dots. And this is actually how the two normal animals interacted with the high stereotypical animal on the left. And you see there was nearly no interaction at all, so they kept to themselves. And on the right hand was how the two high stereotypical animals actually interacted with a normal one, which was much better. So from this figure, it's apparent and very clear that these animals somehow, whether it's by an olfaction or auditory signaling or with sight, actually perceive another animal as being different, and this was quite striking data. The next example of why it's important to look at minorities comes from the marble burying test, in which we saw that in spite of stereotyping responding to escitalopram and large nest building responding to escitalopram, high burying behavior remains completely refractory. It's equally persistent, it's equally needless, it's equally repetitive, it's equally needless, just like nest building in lab conditions and just like stereotyping. But in contrast to those two, high marble burying actually do not respond to escitalopram. And I will elucidate a little bit to this in a few slides from now. If we did not separate these 4 animals from the other 32, you would end up with an average bar on both of those. And you wouldn't have been able to say that marble burying, in essence, does respond to a psychiatric pharmacotherapy or not. Because normal burying behavior, like we would expect, was not responsive at all. So the so what of this will follow in a minute or two. The next application of separating animals in the appropriate way between normal and abnormal came from the escitalopram treatment data of the large nest builders, when we saw that actually normal nest building, the 17 animals that engaged in normal nest building, the behavior didn't alter over time, and it also did not alter as a function of escitalopram treatment. However, in this case, large nest building, the black bars actually attenuated over time and as a response to escitalopram. This kind of supports the behavioral subjective separation between these two populations, because you now have neurobiological data or rather pharmacological data that actually supports a distinct underlying architecture in these behaviors. The fact that burying behavior did not respond to escitalopram amused me quite a lot, and I wanted to get to the bottom of it. And we theorized that it's probably founded in reward and punishment related feedback. So I went to psychological research of the Figea group in the Netherlands, and we started to look at their theories about how patients with specific phenotypes of OCD actually present with reward and punishment related behaviors. So we said, okay, if it's founded in reward, let's see if dopamine is the founding, is the distinguishing construct in these animals. And remarkably, it was. It sounds too good to be true. And remember, we couldn't work with only 4 high marble barriers. So my students still hate me to this day. They never came back for PhDs. I let them screen 400 mice to get about 40 that express high marble burying behavior. And the sandbags is very heavy. So they told me, Prof, never again, please. I've got news for the new ones because it's still, and it tickles me. And what we found was that as opposed to large nest building, high marble burying actually responded to a pro-dopaminergic intervention. Nest building responded to the classic anti-compulsive intervention of high-dose SSRIs combined with a low-dose flupentixol or a risperidone or something of the sorts. High marble burying stopped when we administered rasagiline, the monoamine oxidase B inhibitor. Equally needless, equally persistent, equally repetitive, but it responds to a pro-dopaminergic intervention. With the whole hoo-ha nowadays of the gut microbiome, we contacted colleagues because bear in mind that in South Africa, you don't get the infrastructure always that you need to do these high-tech biomolecular analyses. And so we started to work with a few colleagues in Cape Town and abroad. And what we did in this study is we simply, again, separated animals in normal and large nest building phenotypes. We took poop samples, we sequenced the gut microbiota, and we actually found that if you look quite naturalistically at these behaviors, the red dots would be your large nest building animals, that they present with quite a markedly different gut microbiome composition compared to normal nest building animals. Once again, same rearing conditions, everything, which just exemplifies that the separation and looking subjectively at your data is sometimes quite fruitful. Then our interest in parallel moved to the cognitive foundations of this behavior. And I wanted to ask whether these animals in this lab of ours show some form of risk-taking behavior. So like in humans that express these behaviors, do they persist in spite of being punished? So do they persist in spite of being provided with negative feedback? So not neurobiological in this sense, but more on a neurocognitive level. So we gave them nesting material in lever dispensers and we allowed all the animals. So we first quantified the number of levers that the normal nest building animals actually executed to get to a number of or an amount of cotton wool that actually can build a normal nest. And we set that to a target. And then we allowed all animals to press a lever up to that point. And from that point on, we started to electrocute the levers. And by high or low water, the normal animals immediately stopped their behavior. So we gave it a little bit a lower threshold. So if the normal threshold for building was about 15 lever presses, we allowed these animals to press only for 10. To make sure that your normal nest building animals actually stop due to the electric shock and not due to the satiety of the nesting. The large nest building animals actually persisted in getting shocked to get access to this nest build. We followed this up with work. It was too good to be true for me, so we developed a new behavioral paradigm in which we gave animals a choice between a light and a dark compartment, and we gave enough wool in the dark compartment to build a larger nest, and we really made an anxiogenic big open field in which they had to go into to get more wool. And we actually saw that large nest building animals are actually more risk prone. They take bigger risks in order to get the cotton wool, and that was basically how the setup looked like. So this was done over 3 evenings every time. We tracked this for 12 hours using ethovision. And what we saw on the left-hand side is a figure of a large nest building arena on the morning that we took out the animals. So you will see a small steel container in the bottom left-hand side of this figure, and it's empty. So this animal actually engaged in this open field to get more wool. And on the right-hand side, you have a normal nest-building animal. So he was satisfied with his little bit of cotton wool in the dark compartment. He refused to go out into the white area. We had mirrors all around because that's quite anxiogenic for mice. It's not a form of social enrichment. And that animal simply went out to explore the cotton wool, but he didn't utilize it. He settled for less inside. So in conclusion, I think it's a harsh realization to find out that you wanted already to start to apply a specific animal model for a specific condition, and you are still caught up after 15 years to ask yourself whether certain behaviors really are abnormal. And this is actually what our lab is doing. And to that sense, individuality in animal behavior is really important. Not only to look at different subsets of behavior, but really look at that one animal or two animals that really do something different. The second point I want to make is that in animal research, your behavioral analysis will always be subjective. Look, we look at animal behavior and say to ourselves that when looking at it, this looks a bit different. And there's a big positivity in there because I still believe in the robustness of human evaluation. I just learned in my career a few hard lessons of methods that I wanted to replicate and simply couldn't. And in that sense, the subjectivity of what we are doing can actually be a very bad thing. So when working with animals, you really need to be very honest and very open, and really ask pertinent questions that can be answered in the right way. The last thing I think I want to convey is that when we read papers and we build hypotheses, it's important to accept that if we look at broad samples of animal behavior only over time, then we might miss the really striking, distinct, and accurate data points that might be hiding within that animal's behavior. And if we separate it the right way, we can actually start to ask fantastic questions. And ladies and gents, with that, I think I'm done. I want to thank you again for the opportunity. You are open to email me with any questions. We are always open to help other laboratories or to collaborate with researchers with equal interest than ours. It was an honor to do this for you. It's getting dark and very cold in South Africa. I hope where you are, it's actually becoming warmer and lighter as the day go on. Thank you. Thank you, Sarah, and thank you to the team.
Fantastic. Okay, so the first question that we have here is for you, Jason. Someone has asked, did you see a difference with a round or a squared EOF?
Yeah, so we didn't actually use a square elevated open field. We just used a round one. And so that's a good question. And I think that a follow-up would be to look at Conduct Science's 3D or their elevated open field and see if there are any differences between the two.
Okay, fantastic. Just as a follow-up on that, what differences would you actually expect from that?
I'm not quite sure. I mean, the thing is, typically with walls and Thigmotaxis, the animals like to hide in the corners, but without walls, I'm not sure what that would do. That could be kind of interesting. We have a long-standing debate that I have with many of my colleagues on the other side of the pond when looking at zebrafish behavior and looking at larvae of square versus round wells. And I'm always a bigger fan of round because it encourages just natural movement instead of having corners and having to deal with corners. But I think it's an interesting question that would be something worth following up on.
Okay, awesome. The next question here is for you, Devet. Someone has asked if the populations of the deer mice that you used in your experiments were wild caught or if they were bred in the lab.
So the only stock center of the whole of Pyramyscus genus is actually at the University of South Carolina. So it is a wild type model, but it's actually been bred from stock at USC. So that's the whole right in the beginning. A few years ago, they actually caught a few animals from the wild and started their own breeding stock from that. But they have a very robust breeding program. So these are all lab-bred animals.
Okay, fantastic. Thanks for clarifying that. This next question here is for you, Jason. This question is, would the software be able to detect or recognize episodes of seizure in the field, for example, in epilepsy animal models?
Yeah. I did a quick search on Google Scholar to see if anyone has published with that. Most seizure models, they don't use EtherVision for detection of seizure. Theoretically, yes, it should be possible and doable. And actually, if you have an EEG system or some telemetry, you can integrate that with EtherVision as well and be able to pull in that physiological data in with the tracking data. But it would be something worth looking at also within the activity measure, within looking at pixel changes to see if we could quantify that. Again, it's something that I've never seen, but it's something that theoretically should be possible. And if you have some videos and if you would be willing to share, I'd love to try it. I'd love to test it.
Perfect. So the person that asked that question, Jason's contact info is on our website, so you can reach out to him using that. I've got a question here for you, Devet. Someone has asked if there is a difference in the stereotypical behavior and or in the social behavior between genders and in animals of different ages.
So actually, the sex question, no. So there's no difference. It's male and females. In terms of age, most of these stereotypes develop or become full-blown by the age of 9 to 11 weeks. Although the lab of Mark Lewis and Presti in the States at Florida, at Gainesville, did quite a number of neurodevelopmental studies, and they do express these behaviors earlier, but there's no sex bias. What we did find, I'm so glad for this question because the data is in preparation, but we did find a sex-based difference in the way in which the immune systems of stereotypical animals actually respond, where we saw that males were quite distinct in their presentation when you quantify a simple thing like a neutrophil to lymphocyte ratio. So there are differences in sex, not necessarily on the behavioral side of it, but definitely as to how sex interacts with other factors in the body that may deliver this behavior. And that simply supports our recent notion that female and male psychiatry is actually very different things nowadays, but we are busy with it. So thank you for the wonderful question.
Fantastic. Okay, we've got another question here. It's also for you, Devet. First, this person said they really enjoyed your talk, and then they asked, could you comment on the value of the marble burying assay? When I have inbred mice showing me high levels of burying, what are they telling me?
Okay, so there's a few things. First of all, we did a very big review I will send Sarah the link, then you can get it. So the marble burying test, the first question we ask ourselves is, how do we do it? If we do it on a very light substrate with a relatively large particle size, we need to ask ourselves, okay, what does the behavior look like? So is it marble directed? So is it aimed at the marble, or are my mice just physically active? Because if they are, they will burrow and they will run around in these cages and the marbles will settle to the bottom. The third question we need to ask is, how do we quantify it? Because 85% of all papers out there quantified on a 2/3 buried principle. So if your marble is buried 2/3 of its size beneath the substrate, then we regard it as a buried marble. In that sense, I'm asking from my experience, how can you delineate a 2/3 size of a marble if you have a burying substrate with an average flake size of half the marble? And then it can actually become a question of what does an increased burying count tell me? Less so than what does marble burying tell me? And the short answer is, if you do it like that, in 90% of cases, it's increased motor activity. So that would probably be my major conclusion. It would be very fantastic and informative to repeat that test if it hasn't been done in a coarser substrate and a heavier substrate to see if you get the same results. Because then you can use software to compare the distance moved in the arena, because they can't burrow, with the number of marbles that actually so below the surface. Then you have an accurate data. And that brings me by my last conclusion. So let's say it is increased burying behavior. The only conclusion that can come from it is an increased interest in an object that's not reactive for some or other reason. They proposed the marble burying test as a test of anxiety. I've never seen an animal become anxious due to marbles. I've seen an animal showing interest in marbles, and that's something different. But then we need to be very clear as to the question we ask. So I would say to the delegate that it would depend on the nature of your test and then on the nature of your genetic manipulation. Do you expect it to see something in the motor behavior? Then a marble burying test on a light substrate will be fantastic. Or do you expect it to focus attention on something like we seek for in compulsivity? But then you have to use a more denser substrate in order to come to that conclusion.